Good evening. Thank you very much for coming here. I want to summarize not the last 30 years we did psychedelic research in Zürich, but want to give you a short overview what is the aim and goal, what we do. And in short, we do a lot of studies in healthy volunteers, and a lot of studies are going on in patients. So we have a long tradition. In 1949, we have done the first study in depressed patients, and LSD came to the clinic. And in the 50s, then psilocybin came also from Albert Hofmann. So we had two giants at the clinic. One was Leuner, introducing on the right side the term schizophrenia, yeah, and C.G. Jung, well known for deep psychology. So the clinic is in the tension between schizophrenia and deep access to psychoanalytical thinking. So when I started, I was influenced by a number of other things. It doesn't work. Shit. Maybe I do it like this. Okay, at that time when I started 30 years ago, there was a high time with the in the literature with all these different things. I was trained first in psychoanalysis, and but was also very important, interested in biochemistry. So after biochemistry, I went into medicine. So at that time, I joined with Albert Hofmann, and he was since then a good friend until he died. You never should ask me, are you experienced? All the journalists always ask this stupid question. So what I want to talk today is about consciousness. One of the main goals we do is using psychedelics to study the arrangement of information between neurons using all different kinds of technologies. The second thing I want to go through is depression. We have to conceptualize depression. We cannot just study depression without having any ideas about what's going on. If we want to control patients before and after, we have to know, is there something changed with psychedelics? So I go into emotion regulation and the self. And last but least, then in the end, I go into a study we have done in meditation subjects and studied interaction of psychedelics and meditation skills. So what is the problem? The problem is that you look at the brain from outside. So you can measure the brain in all different kind of things he does. You can look at the neurons, at a single neuron. You can look at several neurons using fMRI or EEG. On the other hand, you have the subjective experience, and you cannot get into the patient and feel with him. So you have to ask him. You need rating skills. So my personal mindset is, I believe in neutral monism. I don't believe in supernatural things. Everything is in nature. So we have to use that tools to try to understand the brain. Particularly, what's difficult, and we use a lot of mathematical things here, to map the inner experience upon what the brain is doing. But we start with the experience, and we ask, what kind of arrangement has to be in the brain that is enabling that experience, that does not explain how consciousness comes from. So consciousness has a long history. It came in about here with the cells, the first cells. You have to have an in and an outside. For a self, you have to have an organization. So the cells were quite clever and put up a system and made boundaries. There was like this aggregation of cells here. So that was the first step in going into consciousness. So if information is coming into the brain through the eye, for instance, or all the sense organs, it is distributed throughout the brain. There's no cell detecting your face or you as a whole. Every single cell in different areas has a different job. They look at contrast, at straight things, at colors, at movements, and in the end, the whole thing has to be integrated, possibly in the frontal cortex. So the information that comes into the brain goes into the cortex. This is this big thing here. The human cortex is the biggest thing. All other animals have very small cortices, and it's layered in 6 layers. So information cuts up through the layers and is connected between the neurons, connection. But it also goes down, goes up, goes down, goes up, goes down, and that allows to loop on what's going on. You have to have a reference. You are in the flow, but you need a meta level to observe what's going on, and you fuse that, and then you can reflect about yourself. You have a sense of self. So we call this strange loops, and it goes back to Gödel and Escher, where upper layers talk with deeper layers and going back and forth and back and forth. So you have expectation, and you have a sensory input. And what you can do to that input is you react. The basic thing in nature, in biology, is an output, motor output, or a cognitive output, thinking, or an emotional output, having emotions. So here you see a typical arrangement, how this cortex is. Millions of neurons. We estimate about 100 billion neurons, and each neuron has about 10,000 connections. So to give you a little bit insight, I'll show you a movie. Oops, too fast. How it really looks. It's an incredible arrangement of cells, and the tools we use measure, for instance, fMRI in a cubic millimeter about 1 million cells. So we don't have the information about each cell. We have information about millions of cells working together. We don't know the micro steps in that incredible organization. It's the most complex thing we have in the universe. So I think you got it. So what's interesting for psychedelics, if you go through the layers, neurons and the pyramidal cells in layer 5 have receptors, serotonin 1A, 2A, information is coming in from the raphe, and they have to communicate through synapses. Electrical currents goes along the axons here and stimulates another cells, and vice versa. So there is electrical information transferring into chemical information, and here the psychedelics come in. They hook on the receptors. So we had, in the first run of studies, tried to find out what of the many 12 receptor families of psychedelics, of serotonin receptors, may be important for that. How you do that? You take a glass kind of needle and stick into the cells, suck in, and take out what's going on in the cell. You can look at the receptor membranes, how these receptors are organized. You suck it off and bring it into the lab, do a lot of molecular biology until you get the structure of the receptors, and you can study that, how it's interacting. So here we measure current, the electricity. At the same time, we measure what's going on in the cell and what's going on if you give a psychedelic. So we get information about a little microcircuitry, and you can inject fluorescent dyes, colors, and then it's now the cell is active and is firing. We put in dyes and can show what's going on in the cell, how is it stretched out, how it is connected with other cells. So what Achatanian found out already in the late 70s, that psychedelics excite these layer 5 pyramidal neurons here, and change the excitation. So this a little bit intuitive. People have to think that they are in a different level than normal waking states. So the trick was to find out what of all these different receptors are important in communication with psychedelics, psilocybin, DMT, LSD. LSD goes out with dopamine receptors, not just the serotonin receptors. So we blocked all these receptors over years, and we blocked this, and that led to the theory that the 2A receptor is important. With ketanserin, it hooks into the receptor, and psilocybin has no chance to go into the receptor, and the symptoms go down. You can give it intravenously. If you're on a trip, you inject ketanserin, in 5 minutes people are completely sober. So that's one thing. But downstream, we have information to the GABA system, dopamine system, which is important for reward, so people feel happy. But much more interesting for psychotherapy, for the therapeutic use, is that psychedelics have a downstream effect on the glutamate system, which goes to different receptors and induce neuroplasticity. They change the number of synapses of contacts here, a very new study from Lee, showing that LSD or DMT activates the neurons, that they make more connections. That's possibly the key to the long-term effects of psychedelics. So there's more complexity. 2A receptors are connected to another system. If we activate these receptors, we block the psychedelic effects. If we block these receptors, we induce in animals a psychedelic effect without LSD. So that's a new street to follow in the future. So there will be a number of new psychedelics coming out. So we're working on that. So it's more complex. It's an important question you have to ask. Serotonin, the most important transmitter here, it's not here, who goes to the same synapse here, is not psychedelic. And a number of LSD derivatives we have tested are also not psychedelics, but activate the receptor. So what's the difference? The difference is that in the cell, the psychedelics activate EGR2, but not the non-psychedelics, this one. Only LSD and psilocybin and DMT. So there are different pathways in the cell that get differentially activated. So that's another key to build up a number of new psychedelics in the future. So we currently work on that we try to fine-tune psychedelics. So you can measure that. It's not just a theory, it's a real human. And you look where the psychedelics go. They go to the 2A receptor. And the more they are occupied in frontal cortices and other regions, more in the parietal cortex and in the visual system, the more they are in a state of oceanic boundlessness. What's going on the next day? The next day, if we measure these guys, they had psilocybin in this case. These receptors go into the cell membrane, they retract. So if you take psychedelics repeatedly, the effect goes down. Everybody who has tested that in 1958, it was tested and showed that psilocybin or LSD leads to tolerance. But what's the nice thing is, the next day, your attentional system is enhanced. So we play with that. Where is the window where the psychedelics are optimal, active, for therapeutic use? Is it acutely, or is it possibly better you learn something the next day when you are much more here in the moment? So that's a question we want to use. I mean, if we work with patients, when is the best window to reintegrate what they have experienced? So the take-home message here is there's one receptor, the 2A, there's the glutamate system, there's neuroplasticity. We have to study the window. When is it really optimal to study these guys? And there's possibly effect on the reward system. Now let's go to depression. Depression is an illness where people are sad, do not move too much, are inside, do not communicate. And one trick is to ask, how can we measure that? It's just a behavior. Can we approach that? So what we know is that depressed patients suffer from rumination. They think negative about themselves, less self-esteem, and they react very strongly to negative stimuli. For instance, negative words or negative faces. So we use first normal healthy. We presented all different kind of faces very fast, flashed out the brain, where is it active, and then you take a psychedelic on all different kinds, and you study how can you influence that negative processing, and then you go back to, for instance, animals and study new psychedelics, which one is most effective. So here is such a task, 3 different kind of face balances, happy, neutral, angry. People are in the scanner, look at the fixation cross, the first target. We give non-conscious, only 10 milliseconds. They just see a flash. They don't realize what's going on really consciously, but the brain has catched the image and is processing that. So this is a deep question. Everybody says psychedelics open up to the unconscious. Here we measure that. It's really so that if you flash people with all different kind of information, they cannot see, the brain detects that, non-conscious. What does psilocybin do here? It reduces the information processing of fearful faces, not for neutral, not for happy. Now you can say why we have a conscious experience. If you do that, do the same, intermingled, you show it longer, 200 milliseconds. People see these faces, and you see the response. Negative faces go down, neutral not, happy also not. That's this. As a consciousness and attention to information adds processing, and that's really good for the idea we had. Can we mix, for instance, mindfulness training and psychedelic therapy? Is there really a reason for that, or is it just fantasy? So we went on, we could show what areas really are decreasing the information for processing of negative faces. We see that, particularly in the amygdala, and mood goes up. And there's a complex network who is doing that for you. But it's not just unconscious, it's a combination of conscious and unconscious, and that's the value of consciousness here. So psychedelic modulate emotion processing to the positive. We did the same with ketamine. Ketamine is a hype too, but ketamine flashes out positive emotions. It reduces that, and you get really kind of, oh, la la, it's not really nice. So let's go to the self. It's much more complex. To assess the self, we heard already from my previous speakers, we can measure oceanic dissolution, negative dissolution, and visual things. We did that with all different kind of inducers, drugs, meditation, sensory deprivation, and so on. So what we want to know is more detailed, what kind of experience drives the psychedelic experience. So this oceanic boundlessness is the mystical scale, has alterations, a feeling of unity and a feeling of ego loss. So these are the most important. And then people have the experience of sacredness and positive effects. They all together drive the experience, but which of them is really important? So the other thing what depressed patients have, they loop in their negative state, and if we lose that self-boundaries, we take away these areas or this information processing that is related to self-referential processing. And we open up more social interaction, feeling of oneness, and we reduce that negative looping in that we take away body experience and self for a few hours. So what we find here is really that with increasing time, when we give psilocybin, we find alterations how brain areas communicate. Some communicate better together, some less. And specific areas go with this oceanic boundlessness. The more boundlessness you have, the more you forget your body and your direct input for the sensory organs. We can go a little bit deeper and we can map how is that with the body. I don't go into these difficult things here, but a number of regions give you a minimal consciousness of your body. And if we look what are psychedelics doing in the communication of that, we found out that it reduces very basic intrinsic systems that give you a sense for inner feelings, what's going on with your body. And if that goes down, that feeling, you open up and you become aware of a much larger consciousness, which is a feeling of being connected. So self-boundaries go down, self-referential processing goes down, and so on. Now, we wanted to use that information, going into the mountains, doing some retreats with Zen masters. They were trained for 6,000 hours at least, had a number of retreats. We had 40, 20 got psilocybin, 20 not. Another retreat we did simultaneously EEG recordings, and in one group, before and after, we looked at neuroplastic changes. So we looked also a lot of properties of these guys. How do they regulate their emotion? And this is the most important. You can suppress emotions if something's going through your mind. You can reappraisal it, you give them a new value, or you accept it non-judgmental. And we measure that every day when they meditated. On day 4, half of them got psilocybin, half not, and we looked them in what state do they go using different scales. The mystical scale was important. Then we measured brain activity after that, 2 days before, and we followed them up for 3 to 4 months and wanted to know, are they changing in behavior? Is the combination of attention here, enhanced attention in meditation plus drug, is that a synergistic interaction? In fact, you can meditate under psilocybin and it goes up versus the placebo group from day to day, but they jump up. Similar mindfulness depth. It was not really significant, but this wasn't so the question. We wanted to know what's with the meditation depth. Here you see red, what these guys have. They have an oceanic boundlessness up to almost 90% compared to placebo. They also had visual things, not here. They have no anxiety, just quasi nothing. Okay. Interestingly, some of the placebo guys got in these states, like the psilocybin people, but you see they're much more dense. Almost everybody went over a threshold of 100, which we set as cutoff to say that's a really strong mystical experience. And here you see these guys here, these 4 guys are similar, but there are differences between placebo, people meditating, and if you add the psilocybin. So meditation skills and mindfulness depth goes up after the retreat. And if you look in the lab, giving the very same dose in lab people, they don't have that extensive blissfulness, this feeling of unity. They are down here green. So the green guys are not so, they have more visual things, and hallucinations are about the same, but they're quite different between psychedelic plus mindfulness meditation. So that you get an idea that this can be done really safely. I'll show you last video, that you get a little bit of an impression how we do this. So they get blinded, nobody knows what people get here. They meditate every day 6 to 8 hours. They were not allowed to speak 5 days. They worked together, they had lunch together, but it was really silent. It was guided by my friend Vanya Palmers, who just gives a talk in another building or room. And it was impressive. They were sitting still, and we didn't know, because it was placebo-controlled, who has psychedelics. So that you can't do with normal volunteers. You have to be really skilled. We do it just with a control group that had not 6,000 hours of meditation, that just are beginners, to tease out the effect of long-term meditation versus beginners. Because patients are beginners, and we do the mindful training also with patients. We give not just the psilocybin, we do the training before and after they got psilocybin to foster the integration. So they do also sometimes after an hour. They cannot sit 6 hours. They make a walking meditation around the center. It's a Buddhistic center. And you see how silent you can be on the drugs. And it's a wonderful area. You have to come to Switzerland and join everybody is invited. So, let's go. Just to the last story, that you believe me, is that the depths of decentering, take it as it is, was the driving force for this mystical experience or for oceanic boundlessness. So it's really important how you prepare subjects. And the other thing is reappraisal. That means if you have negative experiences, and the more you can give them a new value, maybe through talking, through reflection, the less anxiety you have. So that's a good start. I have a little bit too many slides, as always, but that you see that something is going on. After 3 and 4 months, people had an increased acceptance of life, even they were meditators. Self-acceptance went up, concerns for others, less materialistic, spirituality, and a better, how should I say, communication with the end, that life is, has an end, better understanding of the appreciation of death. So I jumped that. Just want to say what's going on. At the moment, we do a double-blind studies in controlled placebo in depressed patients, and we do a European study with 4 countries: Germany, in Mannheim, with Rainer Spannagel. We in Switzerland do the patients. We do crazy things like epigenetics in France, and in Italy we do behavioral study. So I cannot do it without all my students, and I will thank you very much for attention.
We have some time for a few questions.
Yes?
There has been like a new reinterpretation. There's a new book called The Molecule of More about dopamine, and about this process of expanding on the fact that dopamine is maybe not about reward, but it's maybe more about expectation. I would like you to expand a little bit more on how psychedelics would differentiate in their interaction with dopamine in comparison to, for example, stimulants. Like both types of drugs work apparently with the dopaminergic system, but evidently the results are very different. Why would you say so that this happens?
We have compared psilocybin and amphetamine dopamine system, pushing that, and we thought that basically we went up crazy high doses, up to 100 milligrams of amphetamine, 10 milligrams every hour. After 70, people become paranoid and freak out. What we see in the scanner is that there's just activation of the basal ganglia of systems that push more this paranoid thinking, like in schizophrenia. It's really similar. So psychedelics push the dopamine system slightly. We measure that as not much, and it really is nice to the more euphoria. You see that. But stimulants do not stimulate the cortex like just amphetamine or methamphetamine, all these things. It's quite different. Next question. Yes. Yeah?
Are these measures of the experience simply done?
I cannot hear you, sorry.
These questionnaires that you administer, are
yes.
done repeated measures within subjects?
Yes.
So is there a concern that they could understand what you're trying to measure in the experience, and that could sort of mislead a little bit?
I didn't got it really. You asking whether people can fill in the questionnaire?
Well, if they know that you're trying to measure oceanic balances.
During the trip?
Yeah.
Yes, we did that repeatedly. But you can do it maybe every 20 minutes, not more, and we reduce the whole scale. The scale has 96 questions, and you have a better scoring if you use more items, that's for sure. If you measure oceanic boundaries through 3 items on a visual analog scale and summed up to some score, it's okay, but statistically it's low. So normally we use 20 items, and here we use about 10. 10 people can fill in 10, make a scratch on a line 10 centimeters and say, my feeling of connectedness, my feeling of melting with something, my feeling of happiness. That's possible, yes. And it really fits the experience, what we saw over time. Yes?
wondering, you are talking a lot about the 5-HT2A receptor.
Yes.
It's one of the most influential in the psychedelic experience for sure.
Yeah.
So the 5-HT2D receptor is shows activation as well, and at least I'm not aware of a lot of information about it. Do you have any ideas about the relevance, interaction?
We have it only from animals. In animals it doesn't do anything. If you block the 2B, nothing's going on. The 2C is also stroken by psychedelics. Nothing's going on. In the animal you have a model. It's not a very interesting model. They just make this head shake, or they have sensory gating problems, so sensory overload that we can measure in animals, like in humans. We did it in both species. And then you see the 2A is important, but what we have done, we have activated 1A, and the visuals go. So you have an LSD without visuals, just emotions and fusion feelings. But you take away the visuals if you activate the serotonin 1A in addition to activating the 2A. LSD does that also, but if you push it more, then you block the visuals. That's very interesting. So that's the idea. Is the visual important for the therapeutic effect? I think not. It's the emotion, it's the self-boundaries, but the visual is nice to have, but you don't need it. Yeah, it's a plus. But some people are anxious if they visualize their anxiety. You know, that's what's going on. They hallucinate their previous experience where they were anxious, and that's not so nice. If you can cut it off, maybe it's better. Yes? Yeah?
So, first of all, thanks a lot for your talk. And Mr. Vollenweider was incredible when you synthesize so many results of so different disciplines.
That's not easy. Yeah. It's only 3 years, it's not 25.
So, I have a kind of a general question.
Yes.
What would you recommend to some new person who wants to start working in your field? For example, in my case, I'm a student.
Yes. I got every day almost this email from all over the world. People like to come to the lab. If we had more money, we have money, we could do more. Yeah, in Germany I think there's a good opening now. I have friends in Mannheim, in Berlin, in Tübingen. They work with ketamine, they will work hopefully with psilocybin. Yeah, I would maybe here ask Henrik which lab may have they have a consortium in Germany now who tries to knock at the door with the authorities. They are still a little bit stiff here. I don't know how to say that, to be nice. But I think it's a change, because when I started there was Effi Gusulis in Aachen. There was Manfred Spitzer in Ulm. There was Leo Hermle. They did mescaline, psilocybin, MDMA. I think we have even done together DMT. There was a high time in about 95 in Germany. So now they are on the honeymoon again, I hope so. Yes, next question. Yeah? Oh, hi.
You said that the difference between the laboratory setting and the setting.
Yes.
But there have not been the same subjects, right?
Not in this case, no. But it was a nice, we just put them in a room, like a living room, and said, okay, enjoy. They had to draw pictures. It was a nice atmosphere. But it was not a group design. It was single subject. Therefore, we have to do a group study.
Did we get feedback? What setting, what kind of setting, or what the type of the setting they might like the most, for example, in the mountains, because the group setting.
The monks have their eyes closed. Sorry. In therapy, you have these shades. So the outside is not that important. It's important who is doing it, how balanced this person is, how good is the trust. Then you need to prepare them to relax, to let it go. For instance, we have 3 different mindfulness programs doing that. listen to your breath. Body scanning is very good. And our patients do that every day, 10 minutes. They learn it in the clinic, but they do it at home before they go to the experience. And they do it after again. That's a, yeah, you can look at the publication. It's just came out these days. Yes?
You said that you might be able to develop new psychedelics.
Yes,
and
we do.
Already possible to say something specific?
No, it's confidential. It's market. I want to get rich. No, it's a joke. Yes, sure. I don't. Yes.
Yeah. So my question is, why do you think that without this revival of new technologies used by psychedelics, we haven't seen any study of psychedelic phenethylamine?
Phenethylamines like DOI or mescaline, for instance. Yeah, mescaline. Yeah, mescaline. In the models, mescaline is very similar. Very similar. It has most affinity to the 2A receptor, not to the 1A, not at all. There's only the study by Leo Hermle and Manfred Spitzer and Matthias Lichty, my former student, now in Basel, who talks here. They do a LSD, mescaline, psilocybin study, all three together. They look at that. So it's too early. Yes?
How crazy are you getting with the epigenetics study? Like, what are you aiming for?
At least in animal, we have some data. We take blood samples, you know, are you a molecular biologist? So we hope to see whether something is going on because BDNF is activated and all these downstream effects. mTOR is really important. We are NMDA receptors. We see that if you do single cell recording. Maybe there is a permanent change of not just from parents to child, but in the trip there is modification of these genes. So, and that will explain the long-term effects. And if you can optimize that, why not?